The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining us today. You've logged on to the Nelson Labs webinar titled Upcoming Changes to Cleaning Validations for Reusable Medical Devices. Just a few housekeeping items before we get started. If you missed one of our webinars or would like to simply refer back to one, you can always find them on the Nelson Labs website on the On-Demand Webinars page listed under Education. You can receive notifications of upcoming live and on-demand webinars by liking Nelson Labs on Facebook or following us on Twitter or LinkedIn. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know every test matters and requires solutions to complex problems to improve patient outcomes and minimize client risk. And now let's get started. Today we're joined by Griffin Kamek, Consulting Study Director for the Healthcare Reprocessing Section at Nelson Labs. Griffin has helped medical device manufacturers validate the cleaning and disinfection processes for their products by recommending the appropriate cleaning and disinfection procedures for their products, by helping resolve test failures, and by offering continued support to medical device manufacturers even after completion of their validations. Griffin graduated from Brigham Young University, Idaho, with a bachelor's degree in biology and started at Nelson Labs in 2014. By participating in the development of guidance documents and standards as a member of the American Association of Medical Instrumentation, he has remained up to date with the current industry recommendations. Now beginning today's presentation, I turn the time over to Griffin.
Thank you, Mike, for that introduction. And I'm happy to be here and to be giving this presentation. It is an exciting time for reusable medical devices and for the upcoming changes and the new guidance for cleaning validations. And I'm excited to talk about these changes and to talk about the new standard that will be coming out, that will be replacing AAMI TIR30, and that will be ST98. And this presentation will just go over those upcoming changes and the new guidance found in this new standard, this ST98. To give a brief overview of reusable devices and to kind of give some background information and reminder of kind of the types of devices that we'll be talking about is reusable medical devices are devices intended for repeated use repeated use on different patients with appropriate decontamination and other processing between uses. These devices can be invasive, non-invasive, or non-patient contacting. So that means these devices can be critical, semi-critical, or non-critical devices. That's another way of looking at it. And these devices can range from devices used in surgery, such as surgical tools, endoscopes, to things that are non-invasive like keyboards or computer monitors, things like that. And so the umbrella of reusable medical device really encompasses a lot of different medical devices out there. Here is just a slide of a bunch of different medical devices that are out in the field. We have, as I mentioned, devices used during surgery, things like drill bits or ratcheting handles. These are going to be your more invasive, your more critical or semi-critical devices. And then you have devices such as blood pressure cuffs or this heart rate monitor, which are going to be considered more non-critical or non-patient contacting even, such as that heart rate monitor is not really going to come in contact with the patient. And so as I mentioned, that umbrella term of reusable medical devices covers a lot of different devices that are out in the industry and out in the medical field. And we'll be discussing the upcoming changes to the cleaning validations for these types of devices. Now, when you have a reusable medical device, again, an overview of how a reusable medical device is processed or decontaminated in a healthcare facility is you have a point of use process, which is going to be the initial treatment to remove and or prevent drying of soil and contamination. After that process, the device will then go through a thorough cleaning procedure, and then it will go through either a sterilization or disinfection procedure. And this will be dependent on the type of device you have, whether it is a critical, semi-critical, or non-critical device. And each of these processes needs to be evaluated and validated, independently and separately from each other. Now, point of use processing is something that is not a part of validation and is left out from a worst case standpoint. But again, each of the thorough cleaning procedure, the sterilization procedure, and the disinfection procedure each need to be validated. And as I mentioned, we'll be focusing solely on the validation of the cleaning procedure and for reusable medical devices and the upcoming changes to these validations. So a little bit of background. Cleaning validations have been performed primarily, they were based on recommendations found in many different documents, such as the FDA guidance document on labeling and validation for reusable medical devices, which was published in 2015 initially. And then also TIR30, AAMI TIR30 was a major reference document that was used for cleaning validations. However, both of these documents that I mentioned are only reference documents, and TIR30 is a technical report. It's not a standard. So to help provide a little bit more guidance in the industry, it was decided to convert TIR30 into a standard, which will be titled AAMI ST98. This decision stems from the change in the medical device industry that we've seen over the years. So, meaning the increase in new and innovative and complex devices that we're seeing currently is kind of driving this change, and also the need of better understanding of how to perform cleaning validations for these types of devices and other devices. So, out of that need, this standard is being born out of from AAMI TIR30. So a little bit more background on ST98. It has been in development, I believe, for about the last 5 years. So it's been in development for a while, and it's been a collaborative process with the FDA, multiple device manufacturers from around the world, and multiple testing labs as well have been involved on the development of this document, this new standard ST98. And that lets you know how important this standard really is. It has been talked about for the last 5 years. It has been under a heavy review from all of these parties, the FDA, these device manufacturers and testing labs. And so that the recommendations found in there are recommendations based on what is needed for for cleaning validations and ensuring that reusable medical devices can be cleaned and reused between uses. And so it gives you a little bit of background on the importance of this document and kind of all the hard work that has been put into it. So the changes that I'll be discussing, what is new and what is updated in this new standard in ST98 is listed here. And we'll talk about each one of these items in more detail and what that means for you as a device manufacturer, and what that means as well as how that is changing from what is being performed currently. So the first thing we'll talk about is the addition of simulated use cycles to ST98. the addition of controls to cleaning validations with a detailed explanation for each control, the adding of more samples to the positive device control for a total of 3, the addition of an acceptance criteria surrounding the extraction efficiency for that positive device control. We'll discuss the increase in sample size and how test results will be analyzed by applying the standard deviation. We'll discuss the addition of 2 analytes to the list for the evaluation of your cleaning procedure. And then we'll also talk about allowing visual inspection for non-critical devices. So first, we'll discuss the addition of simulated use cycles to the standard. Simulated use cycles were first mentioned in the FDA guidance document for the labeling and validation of reusable medical devices. And in this document, it gave the recommendation and the instruction to perform multiple simulated use cycles to account for the accumulation of test soil over time, over repeated use. However, this document didn't elaborate any further than that, and it was left up to the interpretation of the industry on how to perform these simulated use cycles. How we have defined them to be is we'll take the device through its reprocessing cycle. We'll simulate as closely as we can in the lab to the actual use of the device. So we'll take the device through the surgical procedure and we'll mimic the surgical procedure as closely as possible by applying a test soil and performing a soiling procedure that closely mimics how the device is used in real life. We'll then let that test soil dry on the device, and then we'll take it through the cleaning procedure, whether that be manual or automated. And then we will perform the terminal step, which will either be disinfection and or sterilization. And we'll do that multiple times, usually about 6 times, prior to the cleaning validation. And that has kind of become the industry expectation for these simulated use cycles. Is they're done 6 times, and they are to be performed by mimicking the process that is done in a healthcare facility, the full reprocessing cycle for that device. But again, that FDA guidance document didn't really elaborate, and so what ST98 has done is it has expounded on these simulated use cycles and have further explained how to perform them and what is expected from them. So in ST98, it details how to, as I mentioned, how to perform them, and it details that they should be multiple sequential simulated use and processing cycles to simulate soil accumulation over time. And it also says that these cycles shall be performed for critical and semi-critical devices. So this is an important statement, as we know that shall means a requirement. It's not a recommendation. This will be in the standard, and so it will be expected that simulated use cycles will be performed prior to cleaning validations. Now, what sequential means is that the full simulated use of the device will be evaluated, soiling with allowing the soil to dry on the device, followed by cleaning, and then disinfection and or sterilization using the full cycle of that sterilization. This is similar to what we're currently doing. We are currently performing simulated use cycles in this manner, but now we have a little bit more direction, and we have a reference that we can refer to that will be in this standard now as to how to perform these cleaning validations. Now, something else that ST98 talks about is that the worst case use of the medical device shall be considered, meaning that the full range of motion or actuation should be considered when soiling the device. In addition, you'll need to assess any cauterizing effect, or if the device will cauterize or cut tissue, you'll need to incorporate that into the soiling procedure, as well as if the device becomes heated. If the surface of the device heats up, then you'll need to assess that during the soiling as well, as that may affect how the device is soiled and how the soil interacts with the surface of the device. So this is a further clarification that ST98 is providing that is not elaborated fully in that FDA guidance document. Now, something that was not expounded upon in ST98 is the number of simulated use cycles that are required prior to the cleaning validation. The FDA guidance document does not give a number either. The number that is the current recommendation and is currently we've seen be accepted is 6 simulated use cycles prior to a cleaning validation. This topic was discussed in ST98 thoroughly, and a number of simulated use cycles was added to the document, then removed multiple times, and then it was ultimately decided not to specify a minimum number to perform, and rather you'll need to determine what is the appropriate number of simulated use cycles. So you'll want to look at how complex your device is. If your device is more complex, such as an endoscope or a robotic device, you may want to do maybe more than 6 simulated use cycles. If your device is simple, you may not need 6 simulated use cycles, or if your device is not meant to be used more than 6 times, you may want to look at doing less than that. But again, whatever number you choose will be something that you'll want to discuss internally with your experts and perhaps with a third-party consultant as well, to determine what is the most appropriate number of simulated use cycles to proceed your cleaning validation. Another thing that will be updated in ST98 is that simulated use cycles are not needed for cleaning validations and for non-critical devices rather. So simulated use cycles will only be required for critical and semi-critical devices, but for non-critical devices they are not needed. And this is just based off kind of a risk assessment approach, as the risk for hospital-acquired infections are low for non-critical devices. Now you'll find that there's a little bit of a gray area between non-critical and semi-critical, and not performing simulated use cycles is something that you may want to look into, and you may want to perform a risk assessment just to ensure that simulated use cycles are not required. And you want to look at how your device is used, where it is used, and how much soil the device will see during its use, how much contamination your device will see. And based on that, you will, based on those factors, you'll determine whether simulated use cycles are appropriate or not. But based on ST98, it does say that they're not needed for cleaning validation. So you may not have, they're not a requirement for non-critical devices. You'll just need to, look at your risk assessment and determine whether they're appropriate or not. So the next thing I want to talk about is the changes to the sample size and the controls that will be coming to ST98. And what I have listed here is the current sample size and the current set of controls that we do for cleaning validations right now. And this is based off of what is currently said in AAMI TIR30, and also in that FDA guidance document for the labeling and validation of reusable medical devices. So what we have is a positive device control. This is just one device that is soiled and then unprocessed and extracted. We have a positive sample control. This does not involve a device. It is just an extraction container that is that then has an aliquot of test soil added to it and is extracted. And then for your test articles, right now the current recommendation is to only have 3 samples or 3 devices. And so that is the current industry recommendation. And again, that's based off of some verbiage found in AAMI TIR30 that recommends performing 3 test replicates. And these are devices that are soiled and then they're processed through the cleaning procedure and then extracted and evaluated. The next item we have is the negative device control. This is a device that is not soiled, but it is processed. taken through the cleaning procedure and extracted. And then the negative sample control, this is a device that, or sorry, this is not this is an extraction that does not involve the device. It is just a empty extraction container filled with the extraction fluid. And it serves as a blank for the extraction method or the extraction system. And then currently, if your device is patient contacting, you'll want to also assess for any detergent residuals. And that is typically done through cytotoxicity testing. And this is done with 1 to 3 devices as well. So what we'll talk about is the change that will be coming to the positive device control surrounding the number of samples for the positive device control and the acceptance criteria for the positive device control as well. And then we'll also talk about the sample size for the number of test articles as well, as that is changing in ST98. And then additionally, ST98 has incorporated all of these controls we see here in it, in the document, and with a definition and explanation for each control. So these controls were initially found in the FDA guidance document, the one that I've referenced previously. But ST98 now has these controls. So now we have a standard, or we will have a standard that we can reference to for these different controls. And the standard, ST98, will also provide a definition for each one. So again, the negative device control, that is a device that is not soiled but cleaned. It will be used to determine whether the cleaning procedure interferes with the analytical assays that will be performed at the end. The positive device control, that's a device that is soiled but not cleaned. This shall be used to determine whether an appropriate amount of soil has been applied to the test articles. The negative sample control, this is an extraction that is performed without a device and is used as a blank, or commonly referred to as a blank, and just really used to ensure that the extraction method or the extraction system is not interfering with the test in any way. And then additionally, the positive sample control, which is an extract or an extract without a device that is spiked with a known amount of test soil that is close to the limit of quantitation of the chosen assay. Now, this is important that it's in this document because now we have a list of standards, or sorry, a list of controls in a standard that we can reference and that we can point to, and we can have an explanation for what they are and how they're used. And it gives us a little bit more understanding of how to perform cleaning validations. Now, something else that has been updated for the positive device control is that there's an acceptance criteria for the extraction efficiency for the positive device control. So ST98 requires that an extraction efficiency to be performed for cleaning validations. What this means is that the positive device control will be extracted multiple times. And from those multiple extractions, an extraction efficiency will be calculated. ST98 also gives the requirement, or not the requirement, but the criterion that the extraction efficiency should be greater than 70%. And this was something that has been discussed for years, multiple times. 70% is a number that is, will align with what's found in ISO 15883-5 and other. It also aligned with other USP documents. It's a number that is, often cited as an effective extraction efficiency. And now it will be placed in ST98 as a criterion for your positive device control. Now, if your positive device control falls below 70%, then you'll need to take a look at your cleaning validation and evaluate whether a different extraction method or a modified extraction method would be appropriate in order to achieve a greater than 70% extraction efficiency. So that is something that's going to change. Historically, there hasn't been a acceptance criteria for the extraction efficiency, and so there was no need to assess the extraction method, but now the need is there based on this acceptance criterion. Another thing that ST98 talks about is that the extraction efficiency will be used to calculate a correction factor that will then be applied to the test samples. So your extraction efficiency will be used to correct for the test articles as well. And then for critical and semi-critical devices, a minimum of 3 positive device controls shall be used or shall be performed. This is a major change from current testing parameters, whereas current testing recommendations is only for 1 positive device control. And now ST98 requires for critical and semi-critical devices a minimum of 3 to perform a minimum of 3. And you'll need to calculate the extraction efficiency and correction factor for each of the 3 positives as well. So that's something to keep in mind. And so this is going to change, the number of samples required for testing, and also it's going to change the number of data points generated for your positive. Now, the document does have a caveat, does have a statement that in some instances, a smaller sample size for the positive device control can be used. However, this will need to be scientifically justified. What that means is you'll need to take a look at your device, and you'll need to assess, the risk of your device. Devices that are non-critical, things like keyboards or monitors or carts, things that have a low risk for cross-contamination or hospital-acquired infections, you may be able to perform just 1 positive control for those devices. And so you'll need to, so you'll be able to justify performing less positive controls for such devices, and you'll need to have that justification provided as well, or you'll want to provide that justification up front as well. So another major change to ST98 is the change in the number of test articles that will be tested. So during the development of ST98, a statistician was actually hired, or a team of statisticians were hired to help determine the appropriate sample size or the appropriate number of test articles that should be used during the cleaning validation. What they came back with was a number that was a lot higher than what we're currently doing, and was a number that was very, that was greater than 30. And that was deemed to be somewhat problematic and cumbersome for medical device manufacturers. That is a lot of samples to only be used for a cleaning validation. It is also, that's a lot of material and resources for samples that won't be used for in real life. So what was decided was to not specify the number of test articles, and rather the number of test articles should be determined based on the complexity of the medical device and data reproducibility. So complexity of medical device just meaning, how the device is used, where it is used, how it interacts with the patient, and then also just how complex the geometry of the device is. Are there parts that actuate? Are there, does the device cauterize or cut tissue? Does it come in contact with the blood pathway or with intact mucous membranes or broken skin? These are all parts of the medical device that you'll need to assess to determine the device complexity and to determine the number of samples. Historically, as you saw in the previous slide, is that the number of samples has been 3. Most likely that will increase for critical and semi-critical devices to something such as 6 test articles or 9 test articles. But again, that will be based on your device complexity. It will also be based on data reproducibility and variability, which I'll discuss in this next slide. So what this slide has is a big change that's coming to the cleaning validation process, in which you'll now need to assess for the variability between the data points generated during your cleaning validation. So in this example, which is from ST98, the initial sample size for a particular medical device was selected as 6 data points, and that was based on the device complexity that I talked about earlier. And these are the data points that were generated. Under the current testing recommendations, we would just look at each of these data points and compare it to the acceptance criterion for protein, which is 6.4 micrograms per centimeter squared. And we would just look at whether they're below that criterion, and if they are, we would consider the cleaning validation passing and a success. However, you can look at these results, and you can see that there is a little bit of variability between them, as you have some values that are quite high and some that are low. And so what ST98 requires is that you take the standard deviation of the data points, and then you apply it to the highest value or the highest data point. And if that does exceed the acceptance criteria, then the cleaning validation will be considered failing and not a success, and you'll need to do additional testing. You'll need to generate additional data points and increase the sample size to show that the cleaning procedure is reproducible. Now, this is a big change because, as I mentioned previously, we only would compare the results to the acceptance criterion and whether they're below it or not. But now you have to apply the standard deviation to the highest value, and if that is above the acceptance criterion, then you need to do additional testing. If it's below, if after applying the standard deviation, if that value is below the acceptance criteria, then the data generated will be acceptable and the sample size will be acceptable. So to sum up, the sample size for test articles is going to be based on device complexity. So again, that's how the device is used and the geometry of the device. And then you're going to need to account for variability and reproducibility. So ultimately, your sample size is going to be deemed sufficient if the minimum number of data points are generated per analyte, and that's going to be based off the device complexity. If all the data points fall below the acceptance criteria, and then if there's allowable variation of the results from sample to sample, meaning when the standard deviation is applied to the highest value, is it below the acceptance criteria? If it's not below the acceptance criteria, then you'll have to do additional testing and increase your sample size. Now, with that being said, if enough samples are not provided for testing, or for the cleaning validation, then the simulated use cycles will need to be repeated. So what that means in terms of test article sample size is if after applying the standard deviation, you find that there is the variability is too high, you will need to increase your sample size and redo the cleaning validation. If there are not enough samples that were initially provided, you will have to take those samples through the simulated use cycles again prior to generating those additional results. And then additionally, if, for instance, you determine that your appropriate number of test articles is 6, but only 3 samples are provided for the cleaning validation, you'll have to do the simulated use cycles first for the 3 samples, and then do the cleaning validation, generate the data points, and then repeat the simulated use cycles and the cleaning validation again to generate the next 3 data points for the analytes. This is important because this will impact turnaround time and potentially impact cost, as this is increasing the amount of testing that will be occurring. So that is something to keep in mind as you prepare for cleaning validations in the future, is to plan appropriately for the right number, or for the appropriate amount of time needed to complete the cleaning validation, and to ensure to leave time, or to leave additional time there to account for any test failures, or to account for high variability being there or existing between the data points and needing to do additional testing. So to sum up, the updated sample size and control list is shown here. So this is similar to the slide that I showed earlier, except for the positive device control, we see that there's 3 samples. Again, this is going to be for critical and semi-critical devices. non-critical devices, you may be able to justify only doing one positive device control. And then for the test articles, you're going to need either 3 or more than 3 devices, and this is going to depend on device complexity and data reproducibility. But in terms of the controls we'll be performing, they will be the same, just slightly adjusted in terms of the positive device controls. We'll be performing more. So that is a change. Again, the biggest change to take from this is that the number of samples provided for testing may need to increase, as we'll need more samples for the positive device controls and more test articles for the testing. Or we'll have to do more simulated use cycles in order to achieve the sample size needed. So the next thing we'll talk about is the addition of 2 analytes to the list of appropriate analytes for cleaning validation testing. So this first table we have is the table from ST98 with the list of the different cleaning or analytes that can be used to assess a cleaning validation and their acceptance criteria. Now the 2 analytes that were added are total organic carbon and ATP. Total organic carbon or TOC, is a marker that was researched heavily by the FDA and presented in a webinar. I believe it was in 2005, and but it was not in AAMI TIR30. The rest of these markers, protein, hemoglobin, carbohydrate, and endotoxin, were all listed in AAMI TIR30, but TOC was not. It was only listed in that webinar. But now that it's added to ST98, we will now have again, it will be a great reference point for this marker, and we will no longer have to reference to that webinar as it being an appropriate marker for cleaning. And then ADP, I don't foresee being used for cleaning validations much. I see it more for feasibility or R&D testing, or to be used by healthcare facilities to assess their processing procedures. But in terms of cleaning validations, I don't see it being used often. Now, this list is also kind of listed in, it's listed in hierarchy. So meaning that your protein is going to be the marker that you want to do for your cleaning validations every time. And then the next one you want to choose, will either want to be hemoglobin or TOC, and then carbohydrate and endotoxin and ATP. So you'll want to make sure that you choose the most clinically relevant analytes for your testing. Now, I've added this table from ISO 15883-5. I've added this table to kind of show that the different acceptance criteria that are out there for cleaning validations. Now, the FDA does not recognize alert and action levels. They only have the one recommended level that they need to be below. The alert level that's found in the ISO document it doesn't mean that if you're above the alert level, it doesn't mean that your cleaning validation failed. It just may mean that you need to adjust your cleaning procedure in your IFU when you go to develop your IFU. You may need to adjust it so that it will account for this alert level and hopefully get the protein results below the alert level. Now, if you're above the action level, as the name denotes, you'll need to you'll need to adjust your cleaning procedure and perform additional testing to ensure that you can get the results below the action level. And then on the bottom, I've just put the requirements for critical and semi-critical devices. So critical and semi-critical devices require visual inspection during the cleaning validation, and then a minimum of 2 quantitative analytes. So you don't need to perform every one of the analytes tested. You just need to choose the 2 most appropriate ones. And again, you'll want to choose from this list and kind of choose from the top down. So protein is a marker you'll always want to do. And then your second marker or your second analyte, you'll want to make sure you just choose a clinically relevant one. So if your device sees blood, contact, you'll want to choose hemoglobin. If it doesn't see a lot of blood contact, then maybe TOC would be the next appropriate one. And then carbohydrate and endotoxin may be used, but again, you'll want to just base what you choose off of how the device is used and whether, and choose the most clinically relevant analyte for testing. And then for non-critical devices, visual inspection is the only requirement for those. And this is a change that we'll discuss a little bit as well in a little bit. I just added this in here just to, this isn't a change coming to ST98, but I just added it as for awareness to the different protein acceptance criteria around the world. And so as you can see, there's different requirements based on where you want to market. So the RKI document has the protein requirement as being less than 100 micrograms per instrument. And so if you're wanting to market to places in the EU and to Germany, you may want to look at using some of these other protein acceptance criteria during your cleaning validation. But again, what will be listed in ST98 that was also listed in TIR30 is to be below the 6.4 micrograms per centimeter squared. So the next big change, as I mentioned earlier, is that non-critical devices will only need visual inspection only. So the current way, or the current recommendation for non-critical devices is to perform visual inspection along with 2 quantitative analytes, such as protein and TOC. However, as stated in section 8 from ST98, non-critical devices, so devices whose surfaces contact only intact skin and do not penetrate it, or devices that do not directly contact the patient. These devices should only be evaluated by visual examination of cleanliness, so they don't require any analyte testing. This is a big change as non-critical devices were being held to a similar standard as semi-critical and critical devices, which is somewhat of a high standard for these types of devices. Devices such as blood pressure cuffs and carts and nurse carts or heart monitor carts. These are things that are being held to the same standard as surgical equipment. But what will be in ST98 now is that these types of devices only require visual inspection rather than the 2 analytes. And this will align with what's in ISO 15883-5, which states that for non-invasive medical devices, only visual examination shall be required. And then also what we find in ISO 17664-2, this also is a document for non-critical devices, and it states that you need to validate one method for cleaning, and visual inspection is what is required. And then additionally in ISO 17664-2, it states that disinfection may be carried out concurrently with cleaning of the medical device, meaning that your cleaning validation may be combined with your disinfection validation for non-critical devices. So this change in ST98 is just aligning with what's in with what's currently in other ISO documents as well for non-critical devices. So the overall changes to cleaning validations are just listed here. So an AAMI TIR30 in what's currently being recommended is to do 3 test samples or 3 test articles, 1 positive device control with an extraction efficiency. You compare the test results to just the acceptance criteria, and then analyte testing is performed for non-critical devices. And then what's changing with ST98, that's currently in draft, and will change kind of the testing in the future, is you'll need to possibly do more than 3 test articles. This determination will be left to the complexity of the device and also the data reproducibility. Additionally, 3 positive device controls, each with an extraction efficiency, will be required. And then additionally, there's that acceptance criteria of the extraction efficiency being greater than 70%. That will be added to the ST98 as well. The test results will be compared to the acceptance criteria with the standard deviation applied to the highest value to evaluate the variability between the data, the data generated. That is a change from what we were previously doing, and then ST98 recommends that recommends that no analyte testing to be performed for non-critical devices, which again is in consensus with those ISO documents. So those are the main changes, the overall changes that are coming to cleaning validations in the near future. ST98, as I mentioned, is currently in draft, but we do expect it to be published soon, and so I would expect these changes to be applied soon and to prepare for them soon, to prepare for them now, so that when you go to submit your cleaning validation for review, it will be up to date with the current expectations for cleaning validation testing. One more thing that I wanted to bring up that is a change coming to cleaning validations, not necessarily in ST98, is end-of-life testing and whether it is needed. The current industry method is to provide a method or criteria with which a user can use to determine whether a device is still safe to use. However, ISO 10993 does give the requirement that the biological safety shall be evaluated for the maximum number of validated processing cycles by the manufacturer. This is something that is kind of a hot topic right now, end-of-life testing, based off this statement in the ISO document. And it is something that is currently not necessarily being performed. But it does say in 10993, does have the shall, meaning that it's a requirement. But as I mentioned, the current method that is accepted, as it says in MDR, is that information shall be provided to identify when the device should no longer be reused, such as signs of material degradation or the maximum number of allowable uses. And this is also aligns with what it says in the ISO 17664 and the FDA guidance document, is to give some sort of information to provide a mechanism or method to ascertain whether the device has exceeded its use life and to determine whether the device is functioning properly. However, we have seen that there has been an increase in the request to provide a number of cycles, to provide a number of cycles as to when the device will no longer function properly. And in order to do that, testing will be required. And so, we do foresee in the future as well a change in cleaning validation testing is to also, alongside that, perform end-of-life testing, meaning you will determine the number of cycles that the device can withstand. And so, as a part of that, you'll need to determine how many samples are appropriate for your end-of-life testing, how many cycles to perform, up front, and then additionally, you'll need to determine whether you want to add soiling as a component of the end-of-life testing, which we would recommend doing as you're trying to determine, the full use life of the device, which would include the surgical procedure or the soiling procedure. So these are things to keep in mind as you prepare for cleaning validations in the future, is also to look towards end-of-life testing, as this is something that is being discussed heavily in the industry right now and is being looked at. So you may want to look at it as well and determine what your end of life is for your device. And that brings an end to this discussion on the changes coming to cleaning validations as found in ST98 and other documents. If you, for more educational events and resources, you can visit Nelson Labs at our website, and also our sister companies, Sterigenics and Nordion, at their website as well. And I thank you for joining this presentation. And if you have any questions about cleaning validations or even disinfection validations and sterilization validations, you can contact me at my email or at my phone. And you can ask me any questions you have. And at this time, I will turn the time back over to Mike.
Well, thank you, Griffin, for that thorough and informative presentation. I'd like to thank Griffin for presenting, giving us that presentation, and for his insights on this very important topic. I'd like to thank you, the viewer, for attending this session, and we hope that you found it a valuable experience. I invite you, the viewer, to visit nelsonlabs.com to view additional on-demand webinars on this topic and many other topics that we have available. And we hope that you have a great rest of your day. Thank you.